fermentation for the wine. Thank wine for conversation with these relatives in law of mine. Thank these relatives for you, babe. Thank you for the bouncing boy, doing the best that he can do to keep this family entertained as it navigates the treacherous waters of talking small. All this of all.
Good evening, everyone. Welcome to Science on Tap, brought to you by Science on Tap Cincinnati and Community of Reason. Thank you for joining us this evening. Briefly, again, you can always follow us on Facebook at Science on Tap Cincinnati and also on Meetup at Community of Reason. So this evening, our guest speaker is Kelly Crowe. She's Assistant Professor of Biology at Xavier University. To get this right, she has worked in the field of skeletal muscle gene therapy for over a decade, and most recently has worked with NASA Space Bioscience with the NASA Spaceflight Technology Applications and Research. That's a mouthful. The STAR program, as we like to use in acronyms. Kelly asked if you were allowed to drink at Urban Artifact, and we said, yeah. So I think most of you have your favorite beverage there, so please drink up. If you didn't notice, and if you didn't realize, there's now food trucks at Urban Artifact. So the Good Vibes Eatery was there this evening. They made an outstanding cheeseburger. And I think they will be here going forward on Wednesdays. I'm not sure if they're going to be the food truck every time, but I think so. Anyway, tuck that in the back of your head. So if you're going to get here next month or the months after, you should be able to get food in addition to beer. So without further ado, we'll introduce Kelly, whose talk is going to be Innovations in Muscle Research from Gene Therapy to Spaceflight. Welcome, Kelly.
Thank you all. I am excited to be here. Thanks to Jen and Bob and the rest of the team for inviting me. And I'm really excited to be talking to you guys about some of my own research, which kind of touches on these two fields that I find just absolutely fascinating, which is the field of gene therapy, where you can actually use the gene itself to treat a genetic disease, and then also spaceflight. So as I know that you know, in popular media, recently has been a lot of the idea of longer-term spaceflight. But what we don't know a lot about yet is what that does to the human body. And so there's a ton of active research about the biology of what happens to a human when you're in space, because there's radiation, there's microgravity, and all of these issues that are important to understand before we're just sending people up into space, especially longer term. And so my research touches a little bit on both of those areas, and so I'll be talking to you about skeletal muscle and what happens to skeletal muscle in these different conditions. So to start us off here, I have what is actually a picture. Oh, here, I promised them I would do a good job with the mic, and I'm already doing this wrong. Here we go. This is better, look at this. So we actually have right here a cross-section of a skeletal muscle. So basically, if you were to like take the bicep and chop it this way, look this way, so a cross-section, and then stain it and put it under a microscope, this is kind of what it would look like. And so each one of these is a different muscle cell. And so we'll be thinking a lot today about muscle and what happens to muscle in these different conditions. So we're going to start off by talking about the gene therapy side. And we're going to talk about a very specific disease, a genetic disease that I research on, which is called GNE myopathy. And so I'll explain to you about what G&E myopathy is and how it could potentially be treated by gene therapy, and then how my research weaves into that. And then we'll finish up by talking about what happens to skeletal muscle during space flight for humans, and the bit of research that's been done there, and then where I hope to go with my research that would be relevant for NASA. So what I'm going to be talking to you guys a lot about today are sugars on muscle cells. And that's not something that a lot of people spend a lot of time thinking about. And so when you think about a cell, right, you probably think about something like this. We have a cell hanging out here, and then on the outside, we have our cell membrane, right? And so anyone remember off the top of your head what a cell membrane's made out of? We have our high school bio contingent over here. Yeah, like lipids, right? So fats. Yes, you have fats that are surrounding, you guys did great, that are surrounding your cell, separating the inside from the outside, and keeping everything in. There are no more quizzes. I promised y'all that there weren't quizzes, and then I start off with a pop quiz. And so we all kind of think about the cell membrane as being surrounding the cell, and that's about it. But what we don't think about a lot is actually what goes on outside of the cell membrane. And that is a dense network of sugars called the glycocalyx. So in addition to the fats that are kind of surrounding the cell, you have the glycocalyx surrounding the cell membrane. And it's really hard to visualize because once you kind of take it out of the body, it's pretty unstable. So the cell membrane kind of sticks around, but the sugars are pretty fragile and they break off, and so it's really hard to image appropriately. But if you were to get a really good image, it's easier to see on some cells than others. It might look something like this, where it's a little bit fuzzy on the outside. And the reason why we care about these sugars that are surrounding the cell is because they're actually very important for how the cell functions. Because if you think about it, if anything is heading towards your cell, they don't run into the cell membrane first. The first thing they're going to head into is that glycocalyx. It's the sugars extending from the cell. And so if you're, say, a virus, you might want to attach to that glycocalyx to help you get into the cell. And so the actual type of sugars that make up the glycocalyx, very important for things like recognizing cells, cell-cell interactions, viruses entering cells. All of that strongly altered by the glycocalyx. So this is what I spend a lot of time thinking about, the sugars surrounding a muscle cell. So like I said, we're going to start off talking about a really rare muscle disease called G&E myopathy. And this is the muscle disease that I work on, and I'm working with a bunch of people who are trying to develop a gene therapy for this rare muscle disease, G&E myopathy. And so first off, what you're seeing right here is going to be an MRI. And so you're looking at an MRI of the progression of this muscle disease. So it's a genetic disease, right? The patients who have this disease are born with the genetic mutation that leads to the disease. But I actually don't see any symptoms until like the second or third decade of life, so their 20s or their 30s. And so when the disease starts off, so you can see here in an MRI, this is a cross-section of the upper leg, down here, cross-section of the lower leg. And you can see quite a bit of this darker stuff here, which is muscle. And then the white here is going to be like fat and stuff, and fibrotic tissue. And so you can see that patients, when the onset of the disease happens, there's still a lot of muscle present. But as the disease progresses, you can see that there starts to be a decrease in that muscle mass. So you can see less of that dark skeletal muscle, and then more of that lighter fat and fibrotic tissue. So like scar tissue, basically. And then towards the end of the disease, so this is the quadricep here in the front. You can see the quadricep actually stays pretty spared for reasons we don't understand very well. But you can see almost the entire hamstring in the back of the leg is going to be replaced by that fat and fibrotic tissue. And then the lower leg, there's hardly any muscle remaining. And so in these patients, they usually do lose ambulation. So the patients will use a wheelchair as the disease progresses. So it's a really devastating disease for these patients. And so a lot of the folks that I work with are trying to develop treatments so we can actually do something for these patients. So like I said, it is an adult onset disease, but it's caused by a genetic mutation. So we were actually able to find out what gene is mutated, the very specific mutations that lead to this muscle loss. And the reason why it's called G&E myopathy, myopathy means like a muscle disease, and G&E is actually the name of the gene that's mutated in the disease. So G&E myopathy. And so we're looking at this G&E gene mutation. And I told you I think a lot about sugars outside the muscle cell, right? The G&E gene actually regulates what kinds of sugars go on the outside of the muscle cell. And we'll talk about why that is. So over here we have an overview of what's happening inside the cell and what the G&E gene is doing that changes those sugars on the outside of the cell. So the gene itself, the G&E gene and all the rest of the genes, are hanging out in the nucleus. It's that specialized compartment inside of the cell where DNA is found. And a bit of that DNA is the G&E gene. And the G&E gene ends up making the G&E protein. And proteins can have a lot of different jobs, but this protein in particular has the job of making a sugar, all right? So the G&E protein, once it's made, is going to make this sugar called sialic acid. And sialic acid, as a sugar, is going to get kicked out of the cell and join the glycocalyx. And sialic acid is actually a super important sugar of the glycocalyx. Because remember my fuzzy glycocalyx from earlier? Sialic acid is actually the very last sugar. So it's the terminal sugar. So there's a big old sugar chain coming off, and sialic acid is at the very end. And I told you how important the glycocalyx is because if something's heading towards the cell, first thing they run into is the glycocalyx. Well, actually, the first thing they run into of the glycocalyx is sialic acid because it's right on the end here. And so sialic acid is particularly important as a component of the glycocalyx. So in the case of G&E myopathy, there's a mutation in the G&E gene. So actually, the G&E protein still gets made, but there are mutations in that protein, right? There are little bits of the protein that aren't working quite right. And so what that means for our ever-important sialic acid is that as this gene is mutated, makes a mutated protein, and you end up with less sialic acid than you should have. And so that sialic acid is still kicked out of the cell, the little bit that's left, and it's going to be capping these sugar chains of the glycocalyx outside the cell, but you're going to end up with a lot of those sugar chains that don't have sialic acid on them. So a lot of them are going to be bare like this because less sialic acid is being made. So I wish that I had another slide that told you exactly why that decrease in sialic acid led to that muscle loss. But we don't know. A lot of my colleagues are working very hard on that link, but that's something that's still a gap for us. We're not quite sure. There are a lot of theories that are being tested, but we're not quite sure what it is about that loss of sialic acid that's particularly devastating to muscle. But we know that that link is there. We know that there's this gene mutation, there's less sialic acid, and there's muscle loss. So a therapy, so we can't wait around, right? We can't wait around to figure out exactly what the mechanism is before we try to develop a treatment. But the good news is that with gene therapy, you can actually introduce the gene itself to treat the disease. And so the really cool thing is, even though we don't exactly have pinpointed what it is about that sialic acid loss that would lead to muscle loss, if we bring back the gene, the correct gene, it should help with the disease kind of regardless of what that mechanism is. because what you're doing is you can literally treat a cell that has a mutated G&E gene. You can deliver it a corrected copy of the G&E gene. It should make the G&E protein from inside the nucleus, just like the normal gene would, make that protein, and then it can make sialic acid that's going to go out and join the glycocalyx. So we're really excited about the promise of gene therapy where you're actually using a gene as the therapy itself. And G&E myopathy is a great candidate because there's one gene that's mutated, and we know what it is. So if you're interested in delivering a gene to treat the disease, then this is a great candidate because we know what the G&E gene is. You know, you can rattle off the entire sequence of the G&E gene. So it's a great candidate. So actually the hardest part, I told you I've been working in gene therapy for a long time. The very hardest part of gene therapy is not figuring out the logistics of what the gene is necessarily, although that can be really tricky depending on the disease. But the very hardest part of gene therapy is actually getting that gene inside of cells. Your cells don't generally love to take up DNA that's floating around. That would be very alarming if you're sitting there eating your dinner and it's kind of incorporating into all of your cells. That's not a great situation. Normally, your body does a pretty great job of making sure rando DNA floating around gets chopped up, right? But there is a trick that we have for getting a gene into cells well, and that is a virus. Okay, so viruses get a bad rap, especially right now, but viruses are really awesome at one thing, which is getting DNA into cells, right? They're selfish, right? They want to inject their genetic material into cells. That's their one job, and they do it really well. So with gene therapy, scientists can kind of hijack a virus. So you can basically like scoop out the DNA of the virus and insert whatever DNA you want, like the G&E gene, if you're interested in gene therapy, yeah? Yeah, that's a great question. So in order to make the virus that has the non-mutated gene in there, you can actually use what's called a recombinant approach. So you can actually have cells in a petri dish, and you can introduce the nucleic acid. You can buy nucleic acid on the internet. There are vendors that will kind of synthesize nucleic acids for you. And you can put your DNA into those cells in a petri dish in an incubator, and it will actually incorporate into the virus. Yeah, great question. So we love these vectors. So vectors are just delivery vehicles for our gene. And the vector that we're loving the most right now is a virus because it's so effective at getting DNA into a cell. And of all the different viruses there are, we're loving one in particular. And that is called the adeno-associated virus, or AAV. And so AAV, it's pretty small, which is kind of a limitation, but it's a very safe virus. It doesn't cause disease in the normal state, right? Some of us have probably had AAV infections. You haven't noticed because it doesn't cause any sickness. And so that gives you an extra layer of safety when you're looking to develop a gene therapy. So we really like this AAV, or adeno-associated virus, as a vector for our gene therapy. If y'all have any other questions, feel free to flag me. I'm happy to answer as we go. Or at the end, that's good too. Gene therapy has been particularly good so far for neuromuscular disease, and it's had a lot of promise. So there have been a lot of AAV-based gene therapies, in particular for Duchenne muscular dystrophy, which occurs in young boys. A lot of different approaches to try to, because we know it's the DMD gene that's mutated, so you can deliver the DMD gene to help fix that disease. So a lot of people have been working on that. Another one called X-linked myotubular myopathy, And then the last one here, spinal muscular atrophy, has been a very striking success because that has made it all the way through clinical trials and become a therapy. So you may have heard of Zolgensma, which is the brand name for the AAV gene therapy for spinal muscular atrophy. It's a different disease. It happens in young babies. And unfortunately, without treatment, the children don't survive past 2 years of age. So it's just an incredibly devastating disease. But with gene therapy, lifespan has been significantly extended, and it's received this FDA approval, and it's a very promising therapy. And so the G&E myopathy patient community and the researchers are looking at successes like this and saying, okay, how do we do this for G&E myopathy? So now we're getting into where my research comes in, because a lot of fantastic folks I work with are looking to develop this AAV-based gene therapy. When I was in my Ph.D., that's what I was working on. I was working on the actual development side of the gene therapy itself for G&E myopathy. But now that I'm in my own lab, my lab focuses on biomarkers for G&E gene therapy. Because if you think about it, I was talking about Zolgensma, that big success gene therapy for a different disease. In order to get to that point where it was FDA approved, had to go through clinical trials, right? So you have to get that approval. And when you're actually assessing during a clinical trial, looking at a gene therapy, you need to show a lot of things about that gene therapy. In addition to showing, that it actually helps with the patients, you also need to show that it's getting into cells. And you need to show at what dose is it getting into cells. You know how much of the virus you're giving a patient, but how much of that is actually making its way into cells. And that's where biomarkers and my research comes in. Because if a patient in a clinical trial is given this gene therapy, then with my research into biomarkers, we want to be able to take a slice of muscle, look at it under a microscope, just like we saw at the beginning, and say, yeah, these cells in particular have definitely gotten the virus. And a biomarker is some kind of a clue that can tell you whether or not a cell has actually gotten the gene therapy. So, to see what this might look like, this is looking at a different gene therapy at higher and higher doses. So down here, this is a muscle that hasn't received any gene therapy. This is going to be a low dose, and it gets higher and higher and higher as we go up. And what you can see is that there's more and more green as we go up, right? And what you're seeing is a biomarker for that gene therapy. Each cell is like a teeny little circle here. And each time a cell has gotten the gene therapy, this is for a different disease, right? This is what I'm trying to do for G&E myopathy. But each time that a cell in this disease gets the therapy, the cell's gonna light up like a Christmas tree. And so you can say, yeah, that one definitely got the virus. It definitely has that therapeutic gene. But if you just kind of looked at a muscle under a microscope, it's not gonna look like that. You need to know what to stain it for to make it light up like that. And that's the biomarker I'm trying to find. And so what I think is gonna work really well as a biomarker for G&E myopathy is called a lectin. All right, so a lectin is a protein that specifically binds a sugar, okay? And so a lectin can bind, there are different lectins that all bind different sugars. But some of them, we're gonna circle back here, bind sialic acid, right? Remember how those sialic acid levels are changing if you have G&E myopathy or if you don't have G&E myopathy? There are different levels of sialic acid. So what we can do is use a lectin in some cases that binds sialic acid, okay? And you can put a little glowing tag called a fluorophore on your lectin. So anytime that your lectin, so this is just like a research supply that you can add to a cell. anytime it finds a sialic acid, it's going to bind it and light up. And that can be used as a biomarker, right? Because you would expect that to change depending on whether or not a cell has that sialic acid, whether or not it has the GNE gene. Other lectins are a little bit different because while some lectins are binding sialic acid directly, other ones actually bind when sialic acid is gone. So some of them are kind of the inverse. Those, it's like if it's lighting up like a Christmas tree, it's telling you the sialic acid is gone. And so there are a ton of different lectins in both categories. A bunch of these A types that are binding sialic acid directly, a bunch of these B types that are binding those underneath sugars. And what, we'll look at the list in a second, but what my lab is trying to do is say, like, of all of these lectins, which is going to be the best as a biomarker for this disease. So in the case of GNE myopathy, right, if you're looking at this first type of lectin, where there is sialic acid that's being bound by that lectin. In the case of GNE myopathy, where there's a decrease in sialic acid, you would also expect a decrease in the lectin. So again here, every time you're seeing a circle here, that's a single muscle cell. And that's why you're kind of seeing the staining around the cell. You're looking at basically the glycocalyx, the sugars surrounding that muscle cell. And so you can see here that there's less green surrounding the cell. And that's because we've stained with a lectin that binds sialic acid. So in the case of GNE myopathy over here, less sialic acid, less green. And so that is a potential biomarker. The others are going to do the opposite, right? So these B-type lectins that are kind of binding those underlying sugar structures, in the case of GNE myopathy, when there's less sialic acid, you're going to get more green. And that could also potentially be a biomarker. Because in both cases, you're seeing, like, what is it, can you visualize when sialic acid levels change? Because then, at a cell-by-cell basis, you can say, like, did that one get the gene therapy? Did that one get the gene therapy? And actually get an answer there. So I told you that there are a lot of different lectins under both categories here, right? So these are all of the different specific lectins that have been in scientific papers for changing in staining during GNE myopathy, or in models of GNE myopathy or in patients. So as you can see, there are a ton of different lectins here that have been published. So some of them, the ones that say decrease over here, those are the ones that bind sialic acid directly. And so you would expect them to decrease in the case of GNE myopathy. These ones down here that say increase, these three bind those underlying sugar structures. And so you would expect them to increase in the case of GNE myopathy. But in all these cases, these are different lectins that bind different very specific sugar structures that all seem to be changed in GNE myopathy. And as you can imagine, during clinical trials, they don't love fussing around with, well, we have these 8 different things that might work. Let's try them out. No, they want to know, they want to have a good, solid marker that they can rely on, and you can tell whether or not the sialic acid has been brought back to a cell. So what my lab does is tests these lectins under different conditions with sialic acid changes and say which one of these is the most consistent and the most striking in showing differences in the presence of kind of unmutated GNE or the absence of the unmutated GNE gene. Because we're looking for a really solid biomarker that could be used for a clinical trial as a biomarker. So I'm just going to show you real quick 2 experiments that we've done to try to get at that question. We were talking earlier about cells in a petri dish, right? And that's one of the models that I've used. And so we'll talk about these cells that I kind of grow in my lab with a petri dish and an incubator. And you can actually have cells with GNE myopathy, with the GNE gene, and cells without the GNE gene. And then you can stain them with lectins and look for a difference. So that's what we did first. And then we actually were able to look in the muscles of human patients with and without GNA myopathy. And so I'll show you that second. But first of all, let's look in these cells. So this is where we're actually going to see the data of what we found when we did these experiments. So the first lectin we tested is called MAA, and it's named after the plant they got it from. But this lectin is one of those ones that binds sialic acid itself. And so the first condition we looked at were just kind of normal cells, everything's fine. That's our control. The next are cells where we actually took an enzyme and chopped off all the sialic acid. So in that case, we would expect to see a decrease in our lectin, right? We would expect less brightness because the sialic acid's been chopped off. It's flying around somewhere. We also looked at GNE knockout cells. So these are cells without the gene that makes sialic acid. So again, you would expect that to be decreased, right? Because if you don't have the gene that makes sialic acid and you have your lectin that's binding sialic acid, not going to be very bright. And then finally, we have a condition where we're trying to mimic gene therapy. So we have our knockout cells that don't have the GNE gene, and we're actually putting the GNE gene back in. All right, so picture you're my students, right? You're in the lab. You've just done this staining. You've been growing these cells. You have taken the cells. You're staining them with the lectin. You're finally looking at them under a fluorescent microscope. You know what you expect to see, and there's like nothing, right? They all kind of like look the same. So they were very disappointed. It's like, oh, we were so excited. So MAA, if you ask any of my students, not their friend. They were very bummed out on this day. But okay, you can't just call it at one. Gotta try more, gotta try more. And so the next one we tried is called PNA. Different lectin. It's actually from a peanut. So there you go. That's where the P comes from. But same kind of thing. We have the cells that are normal, the cells where we chopped off the sialic acid, the cells that never made the sialic acid in the first place, and then the condition where we brought the GNE gene back to mimic gene therapy. But this lectin binds those underlying sugar structures. It lights up when sialic acid is gone. So here, when you remove the sialic acid or never make it, you would expect to see more green, right? Because it's saying like, hey, sialic acid is gone. I'm binding those underlying sugars. And then when you bring the GNE gene back, you would expect those values to go back down. Okay, so again, my students, fresh off of their disappointment, back in the lab, they're staining, they have their new lectin, they're excited. It went better. So this time, we saw what we hypothesized that we would see. As compared to the cells that are kind of normal, this lectin doesn't have anything to bind to, right? Because the sialic acid is masking down here. But when we're taking the sialic acid away, suddenly lighten up like a Christmas tree, right? And so very excited about PNA as a potential biomarker. And then what's even more nuts is when you put the GNE gene back, you can see that there's a decrease in the brightness. So you can see, hey, that sialic acid is being put back. It's masking all of these underlying sugar structures, and our lectin isn't binding so well anymore. It's not all the way back to our normal condition, but you can see it's a lot less green than in these conditions. So very excited about PNA as this potential biomarker. But there's only so much that you can tell from cells, right? So the next thing that we wanted to do was actually see, and we also tested a lot more lectins. We're not looking at all those today. I know you saw my table with my 7 or 8 lectins. We're just looking at a few today. But the next thing we wanted to do was look at these in actual patient muscles. So these are biopsies from patients with and without GNE myopathy to see if what we found in the cells would hold up when you're actually looking at a patient. So the downside of looking at a patient biopsy is you can't do that last panel without running a clinical trial, right? You can't look at the replacement side. But the benefit here is that you're looking in a kind of a real life situation. You're looking at actual patient muscle biopsies. So here we are looking in these patient biopsies from patients without G&E myopathy and patients with G&E myopathy. So the first one we looked at here is a lectin called VVA. And this is, again, one of those lectins that binds those underlying sugar structures. So you'd expect it to get brighter in the case of G&E myopathy. This not the best day in the lab, right? And this is fine. This is what happens. Sometimes our hypotheses don't pan out, and that was the case with this lectin. When we use this lectin, you'd expect it to get brighter in the case of G&E myopathy, but we didn't see much of a difference at all. You know, it's not working out. When we did this, when we repeated this, each one of these is a different patient, and so you can see that the averages here with this line are about the same. So we're not seeing much of a difference with patients with and without G&E myopathy. So that suggests to us, at least with this model, that VVA, not the most promising lectin for a biomarker for these clinical trials. But you persevere, test more lectins. The next one we tested is called SBA. Again, finding those underlying sugar structures, you'd expect it to get brighter. And in this case, it actually did get a little bit brighter. We're gonna have to put on our super squinters here to see this at all, but if you look real hard, you can start to see some green circles appear in the case of G&E myopathy. And that's because this lectin is starting to bind when the sialic acid is gone. And when we quantified it, when we did a bunch of them and plotted it out, we could see that there was a statistically significant difference. But what you can see is that this is a very wide spread, and that's because the staining was pretty messy. There were a lot of cases where there's kind of a lot of green speckles everywhere. You know, you're expecting to see that nice glycocalyx surrounding each cell, those green circles we've been seeing this whole time. But in a lot of cases, it was like not a Picasso, who's a modernist. I'm not much of an artist. Yeah, we have a Pollock situation here with the green kind of being splashed haphazardly. And in terms of a biomarker, we're not wanting to show that to the FDA, right? This kind of, there's more green, but not exactly where we expected it to be. But we still had that lectin we were really excited about from the cells, right? We still had our PNA that looked so beautiful in our cells. So the next thing we wanted to do was test our PNA. And it looked really nice. So, again, binding those underlying sugar structures, you'd expect it to be more staining. And in the case of G&E myopathy, we could see these beautiful green circles that are appearing in the case of patients with G&E myopathy, indicating that loss of sialic acid. And when we quantified it, we got a statistically significant difference. I'm showing that change, showing that there's increased staining in the case of G&E myopathy. So before we move into space, while we're still here on Earth, what we thought from these studies is that these lectins are, at least some of them, are changing between non-G&E myopathy conditions and G&E myopathy conditions. It looks like they can reflect those changes, particularly PNA. That's the one we're super excited about. And so that could potentially be used as a biomarker for gene therapy for G&E myopathy. So if a patient were to get the gene therapy, you could then take a muscle biopsy and say, okay, these cells have actually gotten the gene therapy. That could be used to help with figuring out what dose could be used. And that could also help with showing the FDA that, yeah, this is working because the virus actually did its job. It actually got the gene into cells. So we're really excited to continue this research, and again, particularly excited about PNA, to show which lectin has that most robust difference in this disease and could be serving as that biomarker. And then lastly here, I want to chat with you guys about one direction that my research is heading, obviously very different from what we just talked about. But this is thinking about that glycocalyx, those muscle sugars, and how they might be relevant to human health during space flight. Because this is what I think about all day, right? I think about those sugars that are surrounding muscle cells. So, in terms of the relevance of human health in space, I'm sure that some of y'all follow various missions to space. And as we are learning more on the technological side about logistically how to keep folks in space longer, we are subjecting them to the space environment for longer and longer periods of time, right? So, for example, might be a little bit outdated, but as of a while ago, we had 10 astronauts on the ISS, so the International Space Station, and you can actually stay in the International Space Station for quite some time. So when I pulled this, this particular astronaut had been in space for 289 days. But that's pretty long, right? That's like record territory. For the most case, when astronauts go up into the ISS, they stay in space conditions for weeks to months, which is still a pretty long time. But as you may have heard, we are also interested in going back to the moon, right? So revisiting the lunar surface. And as part of NASA's plans for visiting the lunar surface, they involve things like building a base camp. They involve building an orbiting gateway, orbiting the moon. So especially when you're starting to think about a moon-based base camp, we're starting to look at much longer exposure to those spaceflight environments. So maybe thinking months to years that humans would be subjected to these space conditions, again, with radiation and changes in gravity. And I mean, if you think about where we evolved, just like here, right, down here with gravity, less radiation, it's a very different environment and a lot of potential health hazards there that we'll talk about in a second. And then we have the biggie, right? Long-term goal, sending astronauts to Mars. It takes a very long time to get to Mars, right? It takes months and months to go to Mars, and so the total time in space would be years for these astronauts. And so understanding human health for long-term spaceflight conditions becomes very important for the health of these astronauts. So what does that do to the human body? The answer is many not very, very good things. And this is true for a lot of organ systems. And a lot of incredible work has been done by NASA-affiliated scientists who are looking not only at what spaceflight conditions do to the human body, but also what we can do to help prevent those things from happening. Lots of really interesting work has been done. But what we know, based on this research, is that microgravity is going to cause fluids to move around, right? So normally you have gravity pulling on the fluids of your body. And so just one quick example of something that can happen during spaceflight conditions is increased eye pressure. So normally you have gravity pulling fluid kind of away from your eyeballs, but in the case of these microgravity conditions, that fluid isn't going to be pulled down by gravity anymore and can put pressure on the eye. So that's a major area of research. There are also obviously psychological issues. Radiation can do a lot of damage, particularly to things like bone, also muscle. There's issues with circadian rhythm, that kind of 24-hour rhythm, the sleep-wake cycle, tons of issues all over the body. But what I'm particularly interested in is muscle, because with microgravity, you're not having those same strains put on your musculature as you would during a typical gravity situation, right? You just don't have the same force on your muscles, a minute-to-minute basis. And so muscle loss is a very huge issue for astronauts. So this is an astronaut who is kind of coming back. I'm a Russian astronaut, and you can see the folks kind of helping him out, partially due to the muscle loss that was experienced by this astronaut during his time in space. And they've actually quantified this during these different missions. And so each of these is kind of a different mission, and they're looking at the changes in how well the knee muscles function. And this is the percent change from their baseline. So if the number's up here, that means it didn't change. If the number's down here, it means that they lost muscle force. And so you can see in most of these missions, and so this is sorted by days, so this is the mission duration on this axis here. So you can see that particularly as you get into those long missions, 120, 140 days, you're seeing like a 30% loss in muscle force. So a really significant loss of muscle strength happening during space flight. And so obviously, NASA is very serious about this. And so the really good countermeasure that's being used right now to try to help astronauts in the ISS is just old school exercise, right? So right now, astronauts are kind of on the ISS, and they get on this little treadmill for a couple hours a day. So to help with the microgravity, they have a little kind of springboard system that keep them down on the treadmill. And they are kind of running on this treadmill, exercising for 2 hours a day. Astronauts up at the ISS are very busy scientists, right? They're trying to do all of these experiments. And so first of all, even with the exercise, they're still losing some muscle mass. I mean, imagine if we all exercised 2 hours a day. We'd all be ripped. But even with their 2 hours a day of exercise, they're still losing significant muscle mass. And it's also detracting from the time that they would otherwise be spending doing their jobs as astronauts. And so something that I'm interested in is what else can we do besides exercise to help preserve that mass? And so one direction that my research is heading is I'm really interested in those sugars and how those sugars might change. Because what we've seen in the literature, in kind of studies other people have published, we've seen that as you lose muscle mass, there are changes in that sialic acid. And so I'm interested in pinning that down and looking for a really clear connection between sialic acid levels and muscle loss, like astronauts would experience during spaceflight. And then if that turns out to be true, you can target, potentially, the sialic acid. You could have an intervention that changes the sialic acid as a tool to help astronauts lose less muscle mass during these conditions of microgravity. And so I'm very excited about this. I have a meeting next week with a collaborator who is sending me muscles from mice that have been up in the International Space Station. So I'm really excited to look at those and see if there are changes in that sialic acid as I would predict that there would be. Because that could be the first step towards looking at developing some kind of an intervention there. Awesome. Well, I want to thank the folks who did all of this work, my lab members over the years, who have done some of this work, a few of whom are pictured there. I also have a picture that I like to use of all of us on Zoom during a lab meeting during the pandemic. I would also like to thank all of my collaborators. A lot of these are on the side of G&E myopathy, but the LEC lab, who provided the cells used in these experiments, then Ashino lab, who provided the muscle biopsies, the Weissleiter lab, who helped with the human biopsy experiment, Steli Matrana-Rosenbaum's lab, a fantastic group working on G&E myopathy, Paul Martin's lab at Nationwide, and then my funding sources, and the G&E patient community, who's a really fantastic group of folks. And I'd also like to thank all of you for coming tonight. And I would be happy to take any questions. That's all. Yeah?
Kelly, outside of the lab,
Yeah?
In clinical trials,
Uh-huh.
how is the gene therapy injected into the patient? Is it directly into the muscle, or does it go in the bloodstream, or
Yeah,
what is that?
that's a fantastic question. So the question is, how do you, in an actual clinical trial, how do you get the virus into patients? Is it intramuscular? Is it through the bloodstream? And the answer is, it depends on the specific clinical trial. So, I know. Muscle is very interesting for gene therapy because you have so much muscle in your body, and it's all spread throughout your body. And so in a lot of these muscle diseases for which you want to develop a gene therapy, it's particularly challenging to get the virus to muscles throughout the body, right? Your upper arm, your lower leg. And so for gene therapy for muscle disease right now, we're moving towards an IV delivery, so going through the bloodstream. Kind of the older gene therapy clinical trials were intramuscular, so directly injected into the muscle, but IV is more common now. But it can be trickier, right? Because IM, intramuscular, is a direct kind of exposure. IV, it kind of has to get out of the bloodstream and get into muscles. So we're moving in the IV direction, but it can be tricky.
Well, the follow-up then would be, if it's through the bloodstream, how does it get to, the calf muscle that you showed earlier and not into my upper arm?
Yeah, so the idea would be that it would spread to these different muscles. So the idea would be that it would infect different muscles because all of these different muscles, with the exception of the quadriceps, that front of the leg that we saw earlier, I'll go over here. These muscles are all affected, and so the goal is to get it to all of these muscles. Because with an intramuscular injection, you're really targeting like a muscle at a time. But with an IV injection, the idea is that with kind of one dose, you could hit muscles throughout the body. But yeah, muscles, I told you that with gene therapy, it's really hard to get virus into cells. With muscle, it's even harder because of how it's completely spread out throughout the body plan. And there's so much of it, right? If you want to get a gene therapy to the liver, like there's a lot of vasculature, and it's in one little spot, right? It's so much easier to hit. But muscle, it's just like everywhere, and it's really challenging. Yeah.
I had a question about the astronaut. So does the microgravity affect like the muscles that don't seem to maybe be affected by gravity, like the heart, the diaphragm?
Yeah, great question. So the question is, what kind of muscles are affected by microgravity, specifically things like the heart and the diaphragm? So with these issues with muscles that we've seen, particularly for the shorter duration, we've seen that skeletal muscle, kind of the muscles of the limbs, are the most quickly affected. And a lot of it is that we don't know how very long-term exposure will play out in other muscles, because it's likely that if you're looking at a trip to Mars, et cetera, that really it's going to be many different systems that are affected. But the most quickly affected, I would say, would be those skeletal muscles. Yeah, great question. Any other questions? Yeah. Yeah, absolutely. So the question is, how does gene therapy compare to CRISPR? So the gene therapy that I told you about today is called a gene replacement therapy. So you remember how we talked about the fact that there's the mutated gene, and you deliver a corrected copy of the gene, but the mutated gene is still hanging out there? So CRISPR is a different kind of therapy that would actually correct the mutated gene instead of adding a second copy. So that's like, let's say you have a machine at work that's broken, right? A gene replacement therapy, like I talked about today, would be you leave the machine there, you buy another one, and set it up next to it. CRISPR would be like bringing in a technician to fix the original machine. And so actually, newer studies have been looking at kind of a CRISPR gene therapy, not for GNE myopathy in particular, but for Duchenne muscular dystrophy. So you can actually use a virus to deliver the components of CRISPR, because it's nucleic acids, right? Deliver the components of CRISPR into the cell. And then that can kind of correct the gene inside of the cell. And so those can be combined sometimes. But both approaches are good. With CRISPR, you have to really fine tune it because it can be a little bit less efficient because you have to rely on the machinery to, not only do you have to rely on the virus to deliver the parts inside of the cell, but then you have to rely on the second step of effectively fixing the mutation. And so we're getting more and more efficient at doing that, but it's a little bit of a harder task from a development perspective. Yeah, great question. CRISPR versus that gene replacement. Anything else? Yeah.
First off, you did awesome.
Thank you.
And now I can never think of myself as anything but like glazed Krispy Kreme donuts.
I love that. Cells as Krispy Kreme donuts. I'm gonna steal that. That's in my repertoire now.
What's made you interested in GNE myopathy specifically? Like, is your area of study?
Yeah, great question.
Or great.
Yeah, so why am I interested in GNE myopathy in particular? So GNE myopathy is a pretty rare muscle disease. And it's a muscle disease that maybe doesn't get as much attention or press coverage as things like Duchenne muscular dystrophy. And so I'm really drawn to kind of the rare disease patient population because these are, and I've met a lot of these folks who have GNE myopathy, and they are just a really incredible group of people who are just so invested in the research side of things and just extremely supportive. And so it's really kind of about the patient community and being excited to work with them. Yeah. Yeah.
Sorry, I think you have
I just wondered if you could say anything more about where the research is in terms of clinical trial to actual use studies.
Yeah, in terms of the gene therapy? Yeah, great question. So the question is where the gene therapy is in terms of clinical trials. So I do a lot of work with a foundation called the Neuromuscular Disease Foundation, the NDF, and they're like the biggest GNE myopathy organization in the world. They do kind of patient side and then also the research side. And so I work closely with the NDF on this research. And so the NDF is pushing towards a clinical trial. And so there's a whole process to kind of get to that first step in humans. And a lot of that involves work in a lab model like a mouse. And so what the Neuromuscular Disease Foundation is doing right now is taking the virus with the gene and putting it into mice and looking at how that's working and looking at the dosage before it can be translated into humans. And so that's the step where we are right now. Yeah. Yeah.
I guess my question kind of fed on what she was asking, but it's like how early can you administer this in a life cycle of a human? But I guess you don't know that yet.
Yeah, so the question is how early can you administer a gene therapy in the life cycle of a human? That's a great question. So it's very disease dependent. In general, with a gene therapy, earlier is better because as you've seen with those MRIs, as the disease progresses, you're getting more muscle loss. And so generally kind of the earlier that you catch it, the better. So it's an adult onset disease. And so that's, for GNE myopathy, there's a little bit more wiggle room because the onset isn't until the 20s and 30s. And so you would ideally administer before muscle loss associated with the disease. So for other diseases like spinal muscular atrophy, which we talked about a little bit, that was the one that affects babies, that's trickier in terms of catching it early because the disease process kind of starts immediately. And so it's very disease dependent. It depends on when that onset is, when you would want to administer the gene therapy. Yeah. 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 How did I get connected with the NASA project? So it's not up here. So like they were saying, I was in that STAR program. And so the STAR program is basically a NASA outreach to scientists who work in relevant fields and saying like, hey, come learn about what we do. Come learn about how your research could translate into NASA relevant fields. And so I took this training course over the course of a year. And so I got to talk to NASA-funded scientists and learn a lot about kind of the logistics of spaceflight experiments and the logistics of working with spaceflight samples, and talk to a lot of folks who've done really incredible work in the ISS and form collaboration. So basically, NASA kind of actively reaches out to the scientific community at large to try to bring people into NASA research, because what they do is very important for human health, for astronauts. And so they want as many pairs of eyes on the problem as they can get, as many people working on this problem as they can. Yeah. Yeah.
As oftentimes happens with like government-funded research, it has other applications. This seems like it would have other applications to people that have muscle loss for other reasons besides going into space.
Yeah, absolutely.
Is there discussion of that at all, or like other applications, or?
Yeah, so the question is, if this muscle atrophy question is relevant for NASA, would it be relevant in other areas as well? And the answer is absolutely yes. There are tons of types of atrophy. If any of you have ever had a cast on your arm, right, you may remember what it was like, your muscle after that cast was removed. That's disuse atrophy. So there are, in aging adult populations, it's sarcopenia is a type of muscle atrophy. So there are tons of different on-Earth applications for muscle atrophy research, and those are definitely in my mind as well. It's fantastic because NASA work on atrophy can translate into these other applications as well. And actually, one of the most important mouse models for atrophy that's used by atrophy researchers for very non-space applications all the time, it was originally developed by NASA. And so there's a lot of interplay between the spaceflight applications and the on-Earth applications. Anything else? Yeah.
I know that now I don't exactly understand how it works, but I know some genetic diseases can be caught via like screening, like in utero, or like there'll be, I guess, percentage chances depending on like the parents and likelihood. Are neuromuscular disorders, are they in that category of things that can be caught via like screening, or no?
Yeah, so the question is about screening for genetic diseases, because some genetic diseases can be screened for and kind of caught before symptom onset. So that's a really fantastic question, whether or not neuromuscular diseases can be screened for genetically. And this is a very contentious question, actually, because newborn screens, right? You screen newborns for many things already. You can't screen a newborn for every potential genetic disease. It would be prohibitively expensive. Some of them are so rare, or that baby isn't in the right kind of population for it, that it would just be prohibitively expensive. You'd need a lot of like blood from the baby. So it's not feasible to screen for everything. And so then the question becomes, which diseases get screened for? And that's where the contentious bit kicks in, right? Because you want to find that sweet spot of screening for the diseases that a baby would be most likely to potentially have. And also, the second side is that you also want it to be something that you could do something about. If you screen a baby for a terrible disease that might develop later in life that you can't do anything about, then that gets into ethically problematic territories, because you're kind of foisting that knowledge without anything that can be done about it. And so there are many guidelines about when you should screen for something, about things like prevalence, like how rare the disease is, but also, is there a treatment for it? And so before there's a good treatment, then generally screening doesn't occur, because even if you do catch it early, not much that you can do about it. And so, for example, spinal muscular atrophy, now that it has that FDA-approved gene therapy, I know folks who are actively trying to get it on the newborn screen, who are lobbying to get that on the newborn screen because they're saying, hey, we couldn't do anything about it before. Now we can do something about it. So we should screen because, as we were talking about earlier, the earlier the treatment, the better. And so it's something that's really in flux in terms of what diseases get in that screen. Yeah, great question. All right, anything else? Awesome, well, thank you guys so much, and thank you for the fantastic questions. I got excited, and I didn't make it very far. But, you know, that's how it goes.
Thanks, Kelly.
Thanks.
Thank you so much. Another thank you tonight is our friends Andrew and Devin from Radio Artifact. They have been streaming our presentations and recording them. You can find those at YouTube and Twitch at the Radio Artifact channel. Next month, Wednesday, September the 7th, here again, we will have Angela Potanek, who's a professor of philosophy and the Center for Engagement in Science, public engagement in science at the University of Cincinnati. She specializes in the philosophy of science. She's interested in how science works. So think of science more as a verb and not as a noun. She likes to focus on, I would say all of us here are science advocates, and she likes to focus on, in part, how to best communicate science to people who are maybe less science literate. It's not just spouting facts and figures, but there's more of a process to that. So that's what she's going to talk about. And I think that'll be fascinating for all of us to learn a better way to maybe communicate all about science. So again, thank you so much for being here. Thank you again, Kelly. And I hope you have a nice evening, and hope to see you next month. Thanks again. Oh, and I was going to make one comment relative to muscle atrophy. As an aging adult, I can attest to muscle atrophy, and it has adversely affected my golf game.
So that's key. Yeah, exactly. Those are the two options. Obviously the greatest. Well,